Hello everyone. Welcome to this NDS session on physiology. Let's discuss very important high yield topics and questions for the upcoming exam. So we have chosen the most important topics from previous year question papers so that it can be a quick series of revision for you for the upcoming exam. I am Dr. Abirami, your physiology faculty. Yes, to start with the first question. Growth hormone is not released in the following conditions. So the question is about growth hormone. To answer this question, answer is glucocorticoid therapy, cortisol. Let's discuss all about growth hormone and then we'll come back to this question. Growth hormone secretion. Growth hormone secretion is highest during which part of the life? Adolescent, because pubertal growth is very fast. So adolescence, highest levels of growth hormone you can see, followed by child growth, children, followed by adults, and growth hormone secretion begins to decrease during old age. During sleep, what is the effect of sleep on growth hormone secretion? NREM sleep increases growth hormone secretion, whereas REM sleep decreases growth hormone secretion. How you can easily remember this? The first stage of sleep is NREM sleep. When you go to bed and when you start sleeping, the first part of the sleep is NREM sleep. At that time, your growth hormone level increases. So as soon as you start sleeping, you start growing. Remember like that. As soon as you start sleeping, you start growing. Because NREM sleep, as soon as you go to bed, you start sleeping in NREM sleep. During that NREM sleep, growth hormone levels are increasing. But REM sleep decreases growth hormone levels. That is about the growth hormone secretion. Hypothalamic control of growth hormone secretion. Hypothalamus produces three major hormones which affect growth hormone secretion. What are they? Growth hormone, releasing hormone, ghrelin, and growth hormone, inhibitory hormone. These are the three hormones produced by the hypothalamus which controls the secretion of growth hormone. What is the effect of each of this? Growth hormone, releasing hormone, and ghrelin both increase growth hormone secretion. They have positive effect on growth hormone secretion. Or from the name itself, easily you can remember this. Growth hormone, releasing hormone. The name itself is growth hormone, releasing hormone. So obviously it will release growth hormone, increase growth hormone secretion. Ghrelin, growth hormone releasing, GHREL, growth hormone releasing hormone. So that is why the name itself is GHREL, growth hormone releasing effect. So ghrelin also increases growth hormone secretion. Growth hormone, inhibitory hormone. This, the name indicates that inhibitory. So it decreases growth hormone secretion. Which is the growth hormone, inhibitory hormone? Somatostatin. Somatostatin is the growth hormone, inhibitory hormone. Or from the name itself, easily you can learn this. Somat means body or body growth. Stat. What is the meaning of stat? Stat means to stop. It is static to somat. Somat means body growth. Statin. Static to body growth. It stops body growth. So growth hormone, inhibitory hormone is static to body growth because it decreases growth hormone secretion. That's why the name itself, somatostatin. Stat means static, to stop. This is the effect of hypothalamus over growth hormone secretion. What are the other hormones which regulate growth? Along with growth hormone, there are few other hormones which also help in growth. Growth hormone helps in growth during your entire period of growth. Throughout, from birth until you complete your growth, growth hormone levels are high. Growth hormone is helping you in growth. The other hormones which help in growth, thyroid hormone and androgens or estrogens, your sex hormones. Thyroid hormones, especially during the neonatal life. First 2 years of life, thyroid hormones are very important for neonatal growth. That is why thyroid hormone deficiency in newborn babies, what is that called as? Congenital hypothyroidism. Congenital hypothyroidism, which is called cretinism, and cretin babies are dwarf. Why? Because thyroid hormones are important for the immediate and neonatal growth. After birth, the immediate growth is because of thyroid hormone. If a baby is born with thyroid hormone deficiency, congenital hypothyroidism, which is called cretinism, it presents with dwarfism. Growth is stunted in cretin babies. What about androgens? Androgens and estrogens, your sex hormones. As the name indicates, sex hormones, they help in pubertal growth. So the levels are high during pubertal period. Maybe from 9 to 19 years of life. Until you complete your pubertal growth, androgens and estrogens are important. So not only growth hormone, along with growth hormone, thyroid hormone and sex hormones also help in growth. That is the effect of other hormones along with growth hormone on growth. What are the actions of growth hormone? This can be divided into two phases. Actions of growth hormone before fusion of epiphysis and after fusion of epiphysis. Before fusion of epiphysis means the bones have not yet fused. So it may be in a baby, in an infant, not a baby, in the fetus, when the before fusion of epiphysis, or in a baby also, in childhood, when the bones have not grown enough to go for fusion. After fusion of epiphysis means in the adults. So before fusion of epiphysis in fetus or in children, after fusion of epiphysis in adults. What is the effect of growth hormone? Before fusion of epiphysis, before the bones can get fused in a baby, in a child, growth hormone increases chondrogenesis. What is chondrogenesis? Proliferation and differentiation of chondrocytes. That is called chondrogenesis. Chondrocytes are cartilage cells. If cartilage cells are proliferating, it will help to increase the height of the child. So before fusion of epiphysis in a child, growth hormone helps to increase the height of a child. After fusion of epiphysis in an adult, now the bones have already fused in an adult. Now it can't increase the height because already the cartilage cells, the bones have fused. Now you cannot increase the height. So instead of increasing the height, growth hormone will start increasing the other effects. Because it can't increase the height, see what all it is increasing. It increases the soft tissue, it increases the size of organs, it increases the jaws and brows, increases the size of hands and feet. These are the effects of growth hormone after fusion of epiphysis in adults. And that is why the disease related to growth hormone. Excessive levels of growth hormone in a child before fusion of epiphysis results in excessive height of the child. That is called gigantism. So if a child has excessive levels of growth hormone, the height would increase, gigantism. Excessive levels of growth hormone in adults. Now this is after fusion of epiphysis. Now the bones have already fused. So there won't be that much of effect on the height, but rather than that, all others will increase. Increase in the size of soft tissue, organomegaly, cardiomegaly, hepatomegaly, splenomegaly, protrusion of jaw, protrusion of eyebrows, increase in the size of hands and the feet. All these changes together is called acromegaly. So an acromegalic patient will have large hand, large feet, large size of the face, soft tissue, jaws are protruding, that is called prognathism, eyebrows are protruding. So rather than the height, if you see all these changes, it is more towards acromegaly, excessive growth hormone levels in adults. What are the other actions of growth hormone? Growth hormone actions are all related to growth. Whatever it's doing, it is related to growth only. It will finally help in increasing the growth. So what are the other effects of growth hormone? You can remember this. Growth hormone helps in growth. For growth, you need proteins. So growth hormone helps in protein synthesis. For growth, you need electrolytes. The growing tissue needs electrolyte. So the electrolytes are moved from the kidneys into the growing tissue. So growth hormone moves the electrolytes from kidneys to the growing tissue. How? By decreasing the excretion of electrolytes in the kidneys. In the kidneys, it decreases the excretion of electrolytes, your salts, and takes these salts to the growing tissue. For growth, you need energy. Growth hormone gets its energy by breaking down fats. So fats are broken down. That is called lipolysis. So growth hormone causes lipolysis. As a result, when fats are broken down, they release ketone bodies. This is called ketogenic effect of growth hormone. Fats are broken down, ketone bodies are produced. So ketone bodies are increasing. This is called ketogenic effect of growth hormone. So growth hormone is ketogenic. Growth hormone uses fats for energy. What is the other source of energy? Carbohydrate. It will not utilize carbohydrates for energy. See, it's not utilizing carbohydrate. Cells are not taking up glucose. The raw material of carbohydrate is glucose. All the cells, they are not utilizing glucose. If the cells are not utilizing glucose for energy, blood glucose will rise. That is called hyperglycemia. So growth hormone increases blood glucose levels. It may cause hyperglycemia. So two important effects of growth hormone. Growth hormone is ketogenic and growth hormone is hyperglycemic. It will cause hyperglycemia. What are the factors affecting growth hormone secretion? What are the factors which increase growth hormone and factors that decrease growth hormone secretion? What are the factors which increase growth hormone secretion? What is the effect of growth hormone on glucose? Growth hormone increases blood glucose level. So whenever your blood glucose level falls, that is called hypoglycemia. So all hypoglycemic factors will increase growth hormone. What are the hypoglycemic factors? Fasting, starvation, exercise. When you do exercise, exercising muscle will utilize all the glucose. And glucagon. Glucagon is one hormone which is increased during the fasting state. So all these can increase growth hormone secretion. Thyroid hormones and androgens. You remember this diagram? Thyroid hormones and androgens, what is the effect? They increase growth. So thyroid hormones and androgens also increase growth. So they will also increase growth hormone secretion. So they also increase growth hormone secretion. Antidiuretic hormone. What is the role of ADH? Water reabsorption. You can remember like this. For growth, you need water. So ADH increases growth hormone secretion. Increased amino acids. More the amino acids, all these amino acids are taken for protein synthesis. And that is a function of growth hormone. Growth hormone helps in protein synthesis. So whenever amino acids are more, growth hormone would take these amino acids for protein synthesis. These are the factors which increase growth hormone secretion. What are the factors which decrease growth hormone secretion? Exactly opposite. If hypoglycemic factors are increasing growth hormone, hyperglycemic factors would decrease growth hormone. Already if your glucose levels are high, there is no need for growth hormone. So growth hormone levels will decrease. What are the hyperglycemic factors? Cortisol is a hyperglycemic hormone. Cortisol will increase your blood glucose. Glucose itself, already if you have more blood glucose, there is no need for growth hormone. And this is called glucose infusion test. What is glucose infusion test? When you infuse glucose into a normal patient, what will happen if you give glucose? Growth hormone will decrease. So this would decrease growth hormone. But if you infuse glucose into an acromegaly patient, what is acromegaly? excessive levels of growth hormone is called acromegaly. So this acromegaly patient is having so much of blood glucose. Already he is having so much of growth hormone. Acromegaly is a condition where you have excessive levels of growth hormone. So an acromegaly patient is already having so much of growth hormone in excess. Even if you infuse glucose, it will not decrease his growth hormone levels because he has so much. So there is no decrease in growth hormone level. Still growth hormone levels are high. Even after infusion of glucose, if blood growth hormone levels are not decreasing, then it is acromegaly, because an acromegaly patient has so much of growth hormone in excess. How much ever glucose you give, there will not be any decrease in the growth hormone level. Still it will be high only. This is called glucose infusion test for the diagnosis of acromegaly. Somatostatin. What is somatostatin? So this is another hormone. Stat. Look at the name. Stat means static or stop. Somat means body growth. It is static to body growth. It will stop body growth. That is why somatostatin decreases growth hormone. Stat means stop. It is static to body growth. It will decrease growth hormone levels. So these are all the factors which would decrease growth hormone levels. So clearly cortisol, you can see it will decrease growth hormone levels. I think that was the question on. Back to the question. Growth hormone is not released in which of the following condition? What about NREM sleep? NREM sleep increases growth hormone. We already saw. Hyperthyroidism. Thyroid hormone increases growth hormone. Adolescents, growth hormone levels are high. Only in glucocorticoid therapy, growth hormone levels are decreasing, because cortisol decreases growth hormone levels. So that is about the effect of cortisol on growth hormone levels. What about the disorder related to growth hormone? Excessive levels of growth hormone. In children, what is it called? Gigantism. In adults, what is it called? Acromegaly. Deficiency of growth hormone. In children, growth is stunted, dwarfism. In adults, see, an adult cannot become a dwarf again. So growth hormone deficiency in an adult, it cannot affect the height because already you are grown. Rather than affecting the height, it will affect the blood glucose levels. Growth hormone increases blood glucose. Growth hormone deficiency decreases blood glucose. So it results in hypoglycemia. Growth hormone deficiency in adults results in hypoglycemia. So over to our second question. Purkinje cells are found in. The answer is cerebellum. What are these Purkinje cells? Look at this picture. This is the image of a Purkinje cell. So Purkinje cells, how do you identify these Purkinje cells? They have a large dendritic network. This is the cell body from which you can see dendrites. The dendrites, they again and again divide into multiple dendritic processes like this. So if you see such a diffuse dendritic processing system, it is a cerebellum, cerebellar cell, Purkinje cell. So with the diagram, with the histology image only, you can identify if the dendrites are dividing so many times to form a diffuse dendritic network, it is a Purkinje cell of cerebellum. Since there was a question on cerebellum, let's cover that topic. Important points related to cerebellum. So this is the location of cerebellum. Maybe here, can I say somewhere behind, this is the location of cerebellum. What is the importance of this anatomical location? It will get the plan from the motor cortex. It will get the movements from all parts of the body below and will match the plan with that of the movement. That is why it is in between the motor cortex and the movements, your hands and your legs, other parts of the body below. Because it will get the plan from the motor cortex above, movements from my hands and legs below, and it will coordinate the plan with the movement. And that is how cerebellum becomes the coordinator of the body. If there is any defect in cerebellum, it results in incoordination. Incoordination is called as ataxia. That is called cerebellar ataxia. Defect in cerebellum results in incoordination, called as ataxia. Let's learn a little more about the parts of the cerebellum. Cerebellum is made up of lateral parts. These are the lateral parts. The lateral parts are called cerebrocerebellum. Why is it called cerebrocerebellum? Because it gets the plan from the cerebral cortex above. From the cerebrum above, it receives the plan. So, planning is the function of cerebrocerebellum. The medial parts are called as spinocerebellum. Why is it called spinocerebellum? Because it connects to the spinal cord, and from the spinal cord to the muscles, and therefore checks for coordination of the muscles, and that's why it is called spinocerebellum. In the center of spinocerebellum, the centermost part is called as vermis. What is the function of vermis? Vermis is the centermost part of the spinocerebellum. So it controls the coordination of centermost muscles of the body. What are the centermost muscles of the body? Yes, your trunk muscles. So this helps in coordination of trunk muscle. Vermis helps in trunk muscle coordination. Spinocerebellum in general helps in coordination. Vermis especially helps in trunk muscle coordination. So that is about the cerebrocerebellum and spinocerebellum. There's another part called vestibulocerebellum. What is the function of vestibulocerebellum? Vestibulocerebellum is made up of nodulus and flocculus, and that is why it is called as the flocculonodular lobe. Flocculonodular lobe. What are the functions of vestibulocerebellum? As the name indicates, vestibulum, anything related to vestibulum means we are talking about the vestibular apparatus. What is the function of vestibular apparatus? Balance. So this helps in balance and equilibrium. Balance and equilibrium. If you can maintain your balance and equilibrium, you can maintain your gait. What is gait? Gait means the way you walk. The walking style is called as gait. Nodulus and flocculus are related to eyeball movements. So this helps in vestibulo-ocular reflex or the doll's eye movement. What is this doll's eye movement? Have you seen the eye of a Barbie toy? If you rotate the head of the toy to one side, the eyeball will go to the opposite side. This is called doll's eye movement. Or have you seen in movies like Annabelle and all, where they show in close up the face of a ghost, a toy ghost or something like that? When the head rotates to one side, eyeball will go to the opposite side. This is called vestibulo-ocular reflex or doll's eye movement. These are the functions of vestibulocerebellum. What about the nucleus of cerebellum? The cerebellum has outer cortex, which has 5 types of cells. What are the 5 types of cells present in the outer cortex? The largest cell, Purkinje cell, granule cell, Golgi cell, stellate cell, and basket cell. These are the 5 types of cells present in the cortex. Deep inside, there is nucleus of the cerebellum. There are 4 deep nucleus of cerebellum. What are the 4 deep nucleus of cerebellum? The mnemonic is from lateral to medial. Doctors eat good food. Actually no, but the mnemonic is doctors eat good food. What does this stand for? D. D for dentate nucleus. E for emboliform nucleus. G for globose. And F for fastigial. So the mnemonic, lateralmost nucleus is doctors eat good food. D, dentate is outermost. F, fastigial is medialmost or innermost. Doctors eat good food from outermost to innermost. D, E, G, F. Dentate, emboliform, globose and fastigial. These are the different nucleus of the cerebellum. So this is the Purkinje cell, and the question was Purkinje cells are present in cerebellum. With the diffuse dendritic connections, you can easily identify the Purkinje cells. Over to the third question. Brunner's glands are found in duodenum. What are these Brunner's glands? You can see in this image. These are the Brunner's glands. They are present in the duodenum. What is the use of this Brunner gland? Brunner gland produces secretions which have a pH of around 8 to 8.9. Highly alkaline secretion, or I can say the most alkaline secretions are produced in the GIT from the Brunner's gland. Brunner's glands are located in the duodenum. In the duodenum, why do you need such high alkalinity? Because the duodenum, when it receives the food from the stomach is mixed with acid rich food. Stomach secretions are highly acidic. When they reach the duodenum, they can cause severe ulcers in the duodenum, peptic ulcers of the duodenum. This acid can erode the duodenum, and that is why, to neutralize this acid, duodenum produces highly alkaline secretions, the Brunner's gland secretion. If the duodenal secretions are not such alkaline secretions, they will not neutralize the acidic secretion from the stomach. So this acid will result in erosion of the duodenum and therefore recurrent duodenal ulcers. To prevent that only, duodenum protects itself by producing the most alkaline secretion of the GIT, Brunner's gland secretion, with a pH of 8 to 8.9. Over to question number 4. During which phase of menstrual cycle are progesterone levels the highest? So the question is about menstrual cycle, considered as a very difficult topic, but actually no. If you put your little efforts to understand the topic, very simple topic, menstrual cycle. So let's do that. First of all, let's understand about the process of oogenesis. To understand menstrual cycle, you need to understand the basic mechanism of oogenesis. In oogenesis, what is oogenesis? The formation of ovum is called as oogenesis. Oogenesis in a female, it starts even before the birth of the baby. In males, spermatogenesis begins only at puberty, but in females, in a female baby, even before the birth of the baby, oogenesis begins in utero itself. First of all, at 15 weeks of gestation, the female baby is inside the mother's uterus. Inside the baby, the fetus, 15 weeks of gestation. The 15th week in the embryo itself, primordial follicles are formed. At around 28 weeks of gestation, meiosis 1 begins, but is arrested in prophase 1 itself. Meiosis 1 starts, but stops there itself. Even before birth, it starts and stops there itself at prophase 1. So at the time of birth of a girl baby, ovary has around 4 lakh primordial follicles. They are ready. 4 lakh primordial follicles are ready. Now when this girl baby is born, at the time of birth, the ovary has 4 lakh primordial follicles. Now the baby is growing, reaches the puberty. After puberty, every month the girl is going to have menstrual cycle. Every month, during every menstrual cycle, every month, one primordial follicle is selected. From that 4 lakh primordial follicle, one primordial follicle is selected every month for every menstrual cycle. What is the fate of that selected primordial follicle? It is being converted into Graafian follicle. So every month, one primordial follicle is selected and converted into Graafian follicle. And inside this Graafian follicle, it contains the primary oocyte. So primary oocyte is present inside the Graafian follicle. So let's say this month one primordial follicle has been selected. It has been converted into Graafian follicle. Inside this Graafian follicle, you have one primary oocyte. Now, during the menstrual cycle, on the 14th day, there is ovulation. Just before ovulation, the primary oocyte completes meiosis 1. Just before ovulation, the primary oocyte completes meiosis 1. When was this meiosis 1 started? You remember 28 weeks of gestation, when the baby was inside the mother only, this was started. But there itself, it was arrested in prophase 1. So the one which was stopped there, arrested there, again it completes the meiosis 1 just before ovulation. So just before ovulation only, first meiosis is completed. As a result of completion of meiosis 1, there is formation of first polar body and secondary oocyte. First polar body and a secondary oocyte. This secondary oocyte is released on the 14th day. That is called ovulation. Release of the secondary oocyte. When this secondary oocyte is released, if there is a sperm waiting there, it will fertilize. Fertilization happens. As a result of fertilization, once fertilization is done, now it completes meiosis 2. On completing meiosis 2, it forms the fertilized ovum and second polar body. So this will happen only if there is a sperm waiting there. If there is no sperm waiting there, this secondary oocyte will be washed away in the next menstrual cycle. I'll quickly repeat this again. Primordial follicles at 15th week of gestation, they begin to form. 28th week of gestation, meiosis 1 begins but is arrested in prophase 1. At the time of birth, ovary has 4 lakh primordial follicles. Every month after puberty, during every menstrual cycle, one primordial follicle is selected, converted to Graafian follicle. Inside the Graafian follicle, there is the primary oocyte. Just before ovulation, primary oocyte completes meiosis 1. And as a result, it forms secondary oocyte and first polar body. Secondary oocyte is released on ovulation. If there is a sperm waiting, fertilization happens. Meiosis 2 is completed. So just before ovulation, meiosis 1 is completed. After fertilization, meiosis 2 is completed to form a fertilized ovum and a second polar body. This is called oogenesis. Now, where is menstrual cycle here? Every month after puberty, one primordial follicle is selected and converted to Graafian follicle. And after that, there is ovulation. So can you understand, just before ovulation, the entire menstrual cycle can be divided into before ovulation and after ovulation. 14th day you have ovulation. What are the changes which happen before ovulation and after ovulation? What do you think will happen before ovulation? Every month, one primordial follicle is going to be converted into Graafian follicle. So that is called as the follicular phase. So before ovulation, what is the phase of menstrual cycle? Follicular phase. Why is it called follicular phase? Because Graafian follicle is being formed. Primordial follicle is converted to Graafian follicle. It's the formation of Graafian follicle. That is why it is follicular phase. So let's discuss menstrual cycle. So primordial follicle, every month one primordial follicle selected, converted to Graafian follicle. And this is called follicular phase of the menstrual cycle. What is follicular phase? Formation of Graafian follicle is follicular phase. Which hormone do you need for this? Obviously, follicular stimulating hormone. Because you are going to form a Graafian follicle, follicle, you need follicular stimulating hormone. So there is increase in the levels of FSH, follicular stimulating hormone. Now this Graafian follicle, it ruptures. That is called ovulation. It is going to release the secondary oocyte. This happens on the 14th day. Just before ovulation, two hormones are increasing, luteinizing hormone and estrogen. That is called LH surge. So during LH surge, increase in levels of luteinizing hormone and estrogen. During follicular phase, FSH. Just before ovulation, luteinizing hormone and estrogen. Now after ovulation, the secondary oocyte has been released. What is the fate of this Graafian follicle in the ovary? Inside the ovary, this Graafian follicle has ruptured, no? So this is going to be converted into corpus luteum, and that is why this phase is called luteal phase. So after ovulation, after rupturing this Graafian follicle, after releasing the secondary oocyte, what is the fate of the Graafian follicle inside the ovary? Converted into corpus luteum. This is called the luteal phase of the menstrual cycle. Luteal phase of the menstrual cycle. So before ovulation, follicle is formed, follicular phase. After ovulation, corpus luteum is formed, luteal phase. Which hormone is produced by the corpus luteum? Corpus luteum begins to produce progesterone. So after ovulation, progesterone levels are increasing. See, before ovulation, FSH is more. After ovulation, progesterone is more. Just before ovulation, LH and estrogen levels are more. Now, what is the fate of this corpus luteum? If it undergoes fertilization, it will form corpus luteum of pregnancy. If there is no fertilization, it forms corpus luteum menstrualis, corpus albicans, and that is washed away in the bleeding phase of the menstrual cycle. No fertilization has happened, it will form albicans. If fertilization has happened, it will form corpus luteum of pregnancy to maintain the pregnancy changes. So now I think you can understand changes in the follicular phase and changes in the luteal phase. We have also learned the hormonal changes that take place during follicular phase, just before ovulation, and the luteal phase. Now over to this graph where you can see the first 14 days. This is the first 14 days. So during the first 14 days, which hormone is increasing? FSH is increasing just before ovulation. So I'll mark the ovulation with a black line. I'll mark ovulation. So on the 14th day, there is ovulation. So just before ovulation, luteinizing hormone levels is increasing, estrogen levels are increasing. Follicular phase, FSH is increasing. After ovulation, progesterone levels are increasing. So the yellow one is the progesterone, the blue one is FSH, the purple one is the luteinizing hormone, and red one is the estrogen. Very clear. Before the 14th day, FSH is more. After the 14th day, progesterone is more. Just before ovulation, just before the 14th day, LH levels are peaking, estrogen levels are peaking. This is called the hormonal changes during menstrual cycle. So we have learnt the ovarian changes in menstrual cycle. What are the ovarian changes? Follicle is forming, corpus luteum is forming. Follicular phase, luteal phase. Hormonal changes also we have done. What are the uterine changes during menstrual cycle? The first 5 days in the uterus, first 5 days is the bleeding phase. The menstrual bleeding happens. After the 5th day to the 14th day, the uterus has to recover from the bleeding. What is bleeding? The uterine blood vessels, all of them have ruptured, the cells have been dead, and they are all washed away. So the uterus has to replace all them. It has to get recovered from this bleeding phase. That is why the uterus proliferates. And this is called the proliferative phase. So from day 6 to day 14 onwards, to look at the changes also, slowly the endometrium is proliferating. After ovulation, this becomes the secretory phase, because the uterus is going to produce secretions. The glands are enlarging, blood vessels are becoming tortuous, uterus reaches the maximum thickness, it has regained all its thickness, and it begins to produce secretions. And that is why it is called the secretory phase. So this is the uterine changes, proliferates and then it produces secretions. That was a simple idea of the menstrual cycle. So the hormonal changes that take place in the menstrual cycle. Let's mark the 14th day. Before the 14th day, estrogen levels are increasing. After the 14th day, progesterone is increasing. And just before ovulation, LH and estrogen levels, it will peak. And before ovulation, another hormone, FSH levels also keep increasing. What is the ovarian cycle? Inside the ovary, before ovulation, follicle is forming, follicular phase. After ovulation, corpus luteum is formed, so luteal phase. What is the uterine changes in pregnancy, in menstrual cycle? First 5 days, bleeding. 5th to 14th day, proliferative phase. 14th to 23rd or 28 days cycle, it's a 28 day cycle, secretory phase. So these are the uterine changes that happen during menstrual cycle. So back to the question. During which phase of the menstrual cycle are progesterone levels highest? After ovulation, corpus luteum begins to produce progesterone. So between ovulation and the next menstrual cycle, menstrual bleeding, progesterone levels are high. That was about the menstrual cycle. Over to the next question. Which of the following is true about secondary active transport? The answer to this question is transport of one molecule dependent on the other. Let's learn about secondary active transport. Then we'll come back to this. So one easy way of understanding transport mechanism is with the help of a mountain. Let's say this is a mountain. And you are here. You want to climb this mountain. You want to go from low to high. So when you go from low to high, you are moving against concentration gradient from low to high. So this is against concentration gradient. Do you need energy for this? Yes, we need energy. You have to climb the mountain actively. So this is called active transport against the concentration gradient and requires energy. What are the types of active transport? How do you get your energy? The primary source of energy is, say if you break your own ATP, get your own energy, that is called primary active transport. So all ATPase is primary active transport. What are the ATPases that you know? Sodium, potassium ATPase, hydrogen, potassium ATPase, calcium ATPase. So all ATPases are primary active transporters. Secondary active transport. See now, you want to climb the mountain, but you don't want to break your ATP. But still you want to climb the mountain. So what you can do, if your friend is already climbing the mountain, you just climb on top of him and he will carry you up. See, now also both of you are going against the gradient. Both of you require energy, but you don't break your ATP. You are getting your energy from your friend. Who is that friend? Most of the time, sodium is the friend. Along with sodium, dependent on sodium, if any other substance is transported, that is called secondary active transport. Example, sodium iodide symporter, present in the thyroid gland, or sodium glucose cotransporter, SGLT, in the intestines and the kidneys. Along with sodium, dependent on sodium, if one other substance is transported, then it is called secondary active transport. What are the different types of secondary active transport? If sodium and the substance are moving in the same direction, then it is called a symporter. Symport means sodium and the substance are moving in the same direction. Sometimes sodium hydrogen exchanger or sodium calcium exchanger. Exchanger means sodium is moving in one direction, hydrogen and calcium are moving in the opposite directions. This is called an antiporter. If sodium and the substance move in same direction, symporter. If they move in the opposite directions, then it is called antiporter. This is about active transport. What about passive transport? In passive transport, you are already on top of the mountain. You just want to come down. Now you are moving along the concentration gradient. From high to low, you are coming from up to down. Do you need energy? No energy is required. You just lie down there, you can roll and you can come down. No energy is required. This is called passive type of transport. Active transport and passive transport. In passive transport, there are different types. Simple diffusion. Facilitated diffusion. Simple diffusion means you lie down there simply and you roll and you come down. That's all. Just like that, you roll and you come down. Nobody is carrying you. Simply you come down. This is called simple diffusion, especially for oxygen and carbon dioxide. The major life-saving gases, they don't have any carriers. They don't have any transporters. Simply they are transported, oxygen and carbon dioxide. Facilitated diffusion. Facilitator. Facilitated means there is a facilitator. Somebody is going to help you. There is a bus on top. The bus is going to carry you down. But the problem with a bus is, a bus can hold a maximum of 50 people. If you are the 51st member on the queue, there is no place for you. When it reaches a T-max, already if the bus has reached its maximum, the transporter has reached its maximum, it will reach a T-max. After that, there is no place for you. You can't come down. It gets saturated. So Tmax and saturation kinetics are followed by facilitated diffusion. If the transporter can reach a Tmax and it gets saturated, it is called facilitated diffusion. Example, GLUT, glucose transporters, UT, urea transporter. GLUT, it will keep on transporting glucose, but at one point if it reaches the Tmax, after that glucose cannot be transported. Same with urea transporter also. Simple diffusion and facilitated diffusion. The other type of passive transport is osmosis, the movement of water. Water can move simply or it can use transport proteins, aquaporins. The other type is of passive transport, non-ionic diffusion. Non-ionic diffusion means not for ions. Then for what? For acids and base. For weak acids and base. Acids like salicylic acid absorption or base like ammonia excretion. All this is non-ionic diffusion. These are the different types of transport mechanisms across the cell membrane. Back to the question, which of the following is true about secondary active transport? Does not require active transport? Wrong. It is an active transport. It allows only co-transport? No. It can allow antiport also. Symporter is a co-transporter. It is a type of facilitated diffusion? No. It is a type of active transport. Transport of one molecule depends on the other? Yes. So this is true about secondary active transport. 2, the next question. A substance which travel down its concentration gradient is known as. Yes, just now we discussed facilitated diffusion. Facilitated diffusion is a type of passive transport. Down its concentration gradient means along the concentration gradient. Antigen present in all ABO blood groups is. Which is that antigen present in all the ABO blood grouping system? First, let's discuss about the blood group systems. Different types of blood grouping system. First one is ABO blood grouping system. According to ABO blood grouping systems were discovered by Landsteiner. This is the RBC and this is the plasma. On the RBC, all the blood groups have this basic antigen, H antigen. On the RBCs, there are antigens, and in the plasma, there are antibodies. All blood groups have H antigen. If you have the enzyme which can convert H antigen into A antigen, then you become A blood group. So an A group person will have A antigen and H antigen. If you have the enzyme which can convert H into B, then you become B group person. So a B group person will have H antigen and B antigen. If you have both the enzymes, then you can convert H into A and B. Now you become AB blood group. So an AB group person will have H antigen, A antigen and B antigen. If you don't have both the enzymes, then you cannot convert H into A or H into B. So you will have only H. This is called O group person. So an O group person will have only H antigen. So basically you see, H antigen will be present in all the blood groups. Based on whichever enzyme you have, if you can convert H into A group. If you can convert H into B group. If you can convert it into both, AB group. If you cannot convert it into both, O group. So this is based on the antigen present on the RBC. What is Landsteiner's law? According to Landsteiner's law, if you have any antigen, the corresponding antibody will be absent. For example, A group person. Any A group person will not have anti-A antibody. He will have only anti-B antibody. He will not have A antibody. Any B group person will not have B antibody. He will have anti-A antibody. AB group person, since AB group person has both A antigen and B antigen, he will not have both A and B antibody. O group person, there is no A, no B antigen, so he will have all the antibodies. So he will have anti-A and anti-B antibody. So what does this tell us? If any antigen is present, the corresponding antibody will be absent. Why? If they are present, you have A antigen, you have A antibody also, both will react, agglutination, lysis will happen, hemolysis will happen. So any antigen you have, corresponding antibody you will not have. Take O group, if you don't have both A and B antigen, then you can have anti-A, anti-B antibody. So any antigen is present, same antibody should not be present. That's all. If antigen is absent, then the same antibody can be present. These are the two Landsteiner's law. There is a special blood group called Bombay blood group. What is this Bombay blood group? See, look at an O blood group. O blood group person has A antigen, B antigen and H antigen. O blood group person has only H antigen. Even if this H antigen is absent, then it is called Bombay blood group. So Bombay blood group person will not have any antigen. He will not have H also. Normally people will have H antigen. All people will have H antigen. If any person is having no H antigen also, then it becomes Bombay blood group. Now can you say what all antibodies will be present in Bombay blood group? Since all the three antigens are absent, he will have anti-A antibody, anti-B antibody and H antibody also he might have. This is called the Bombay blood group person. So this is how Landsteiner's laws were derived. Now back to the question. Antigen present in all the ABO blood groups is H antigen. Yes. What is the other type of blood grouping system? One is ABO blood grouping system. That is what we have discussed. The other type of blood grouping system is called RH blood grouping system. According to RH blood grouping system, if you have RH antigen, which is otherwise called D antigen, you become RH positive. If you don't have RH antigen, you will become RH negative. Simple. If you have RH antigen, RH positive too. So A positive means you have RH antigen. A negative means you don't have RH antigen. So RH will decide whether you are positive blood group or negative blood group. ABO will decide whether you are A or B or O. RH will decide whether you are positive or negative blood group. Both these groups combined, ABO group, RH blood grouping. When you combine both, that is where we get this whole blood grouping system. AB positive. It means AB came from ABO blood group and this positive came from RH blood grouping. Over to the next question. Which is not a respiratory neuron? Answer is caudal ventral lateral medulla. Let's discuss about respiratory neurons and then we'll come back to this question. Control of respiration. Respiration can be controlled by voluntary centers in the cortex and involuntary centers in the medulla. From both voluntary and involuntary centers, impulses come down to the spinal cord. Cervical segment C3, C4, C5. From where you have the phrenic nerve arising. Phrenic nerve innervates the diaphragm. When diaphragm contracts, then you breathe. You do respiration. This is how normally we breathe. So we can breathe both voluntarily and also involuntarily. What are the brain centers? Neural control of respiration. How does brain control respiration? Brain has pontine centers and medullary centers. In the pons, pneumotaxic center, apneustic center. In medulla, pre-Botzinger complex, dorsal respiratory group and ventral respiratory group. What is the role of pre-Botzinger complex? Pre-Botzinger complex is the pacemaker for breathing. And any damage to the pre-Botzinger complex results in Ondine's curse. What do you mean by pacemaker? Pacemaker means it produces the first rhythm for the breathing. That's why it is the pacemaker. Pre-Botzinger complex becomes the pacemaker for breathing. So this is like the SA node in the heart. What is Ondine's curse? When there is any damage to the pacemaker, if this is being damaged, the patient presents with Ondine's curse. This word Ondine came from a fairy tale. Ondine was a little mermaid who was living under the ocean. This Ondine fell in love with a prince. This boy said, "I'll marry you." Finally, he cheated on Ondine. He went and married another princess. Now Ondine got very angry and Ondine cursed him. What was the curse? If you sleep, you will die. What is this curse? Damage to the pacemakers. If the pacemakers are damaged, then pacemaker is lost. So involuntary breathing is lost. So how will this person breathe? He can breathe only by voluntary breathing. When you are awake, you can do voluntary breathing. But if you sleep, you can't do voluntary breathing. The person will die. If you sleep, you will die. Because involuntary center is gone, only voluntary breathing you can do. What is voluntary breathing? Every breath, if you say inhale, exhale, and you breathe like this, that is called voluntary breathing. So if there is damage to involuntary center, only like this, by doing voluntary breathing, he can breathe. When he is awake, you can say inhale, exhale and breathe. What will happen if you sleep? You can't say inhale, exhale. You will lose your voluntary breathing. So if you sleep, you will die. That was a very interesting Ondine's curse. So how will this person sleep? How can you live without sleeping? Yes, whenever you want to sleep, you go to the ventilator, get some artificial respiration, sleep and come back. That was about the Ondine's curse. The other centers, DRG and VRG, dorsal respiratory group, helps in inspiration, produces the rhythm for inspiration. Ventral respiratory group, VRG, expiration but not normal expiration, forced expiration. When will you do forced expiration? When you climb on a mountain, you're running on a treadmill, or you take a deep breath for singing on a high pitch, or when you hyperventilate. Whenever you do forced expiration, VRG is the center. Normal expiration, there's no center at all, because expiration is a passive process, air will move out. But when you do forced expiration, you need ventral respiratory group. What is the mnemonic here? Dive. DI, dorsal respiratory group, inspiration. Dive, VE, ventral respiratory group, forced expiration. Dive. Now what are the pontine centers for breathing? Pneumotaxic center, apneustic center. Pneumotaxic center, this is the switch between inspiration and expiration. It will tell you when to stop doing inspiration and when you should do expiration. It is a switch between inspiration and expiration. And if there is any damage to this switch, you will not know when to stop inspiration, when to do expiration. You will keep on doing inspiration. So breathing becomes slow. Slow breathing. If you keep breathing slowly, you take in more air. So tidal volume increases. Damage to the pneumotaxic center, slow breathing, tidal volume increases. Apneustic center. This can prolong inspiration and it can cause apnea. What is apnea? Temporary cessation of breathing is called as apnea. So these are the brain centers which control respiration. Over to the question. Which is not a respiratory neuron? Dorsal respiratory, yes, inspiration. Ventral respiratory, yes, forced expiration. Pneumotaxic center, it is the switch. Option D, caudal ventral lateral medulla. This is not a respiratory neuron. A 10th question. Infinite feedback gain is seen in. What is this infinite feedback gain? For any feedback control system, to assess the efficiency of a feedback control system, we calculate something called gain. Gain is equal to correction divided by error. What is correction divided by error? If there is any system, if there is some mistake in the system will try to correct itself. How much ever it has corrected is the correction. It can't correct 100%, it leaves some error. So that is the error left by the system. If you divide the correction by the error, you will get the gain of the system. So gain is equal to how much the system has corrected divided by how much the system has not corrected. How much the system has left an error. So gain is equal to correction divided by error. Now let's say if in any case error is 0. The system has not left any error. 100% pakka it's functioning. No error at all. Anything divided by 0. What is the answer? Infinity. If error is 0, then gain becomes infinity, because anything divided by 0 is infinity. So gain becomes infinity. Which is that only system which will have no error? Renal regulation of BP. When kidneys are controlling BP, when kidneys are regulating blood pressure, they will leave no error. So error is equal to 0. Since error is 0, gain becomes infinity. So this is the only system which has infinite gain. Can you understand? There's no error. Error is 0. That's why so much of gain. Infinity is the gain. So infinite feedback gain is seen in renal output. Kidney regulation produces infinite gain. Yes, with that we complete our quick rapid session on the most important MCQs, and more than that the most important topics we have covered. So thank you so much. We'll end this session with this beautiful quote, life is beautiful. Just enjoy life and keep going. And we will see in another session of discussion in physiology. Thank you so much. This is Dr. Abirami, your original guru for physiology. Thank you.